Today we are going to discuss pancreas from physiology point of view. Let us discuss the islet of Langerhans. It consists of many cells like alpha cells, beta cells, delta cells, then F cells. The alpha cell produces glucagon, beta cells produces insulin, delta cell produces somatostatin and gastrin. F cell produces pancreatic polypeptide. Then there are some other cells like D1 cell. It produces VIP. There are epsilon cell. Epsilon cell produces ghrelin. Okay. Now the most important, the one we are going to discuss is insulin. Insulin was discovered by Banting and Best. Before starting with biosynthesis, there are 2-3 things I want to tell you about insulin. First, it is a polypeptide. It is an anabolic hormone. It is a polypeptide. It consists of 2 chains of amino acids. The 2 chains of amino acid, and they are linked together by disulfide bridges. Okay. Insulin secretions, it begins at 12 week of age. 12 week of gestation actually. 12 week of gestation. Okay. The difference, the beef insulin, if you compare beef insulin, pork insulin with human insulin, the beef insulin differs by 3 amino acid from human insulin, while the pork insulin differs by 1 amino acid from the human insulin. Okay. Now let us see the synthesis of insulin. Okay. So let us discuss the biosynthesis of insulin. Okay. We all should know that insulin is synthesized in the ribosome of the rough endoplasmic reticulum. Okay. And it is synthesized as a proinsulin. Okay. And then get translocated into the Golgi apparatus. Then it is get translocated into the Golgi apparatus. Then it transforms into insulin. And here it is synthesized into some storage granules. And the storage granules contains proinsulin, insulin, and C-peptide. We'll discuss about each one of them. Okay. And these storage granules, then they will come in the cell. They will come close to the cell membrane. Okay. And then their membrane will fuse with the cell membrane, and hence the insulin will be exocytosed. It will be released outside. Okay. So this is how the insulin happens. Now, this was just a brief I have told you that how insulin is formed in the ribosome of the rough endoplasmic reticulum. Then it formed in a pre-form. There is a proinsulin. Then it converts into an insulin when it passes through the Golgi apparatus. Okay, and then it is secreted into the storage granules. Then it is exocytosed outside the cell. Okay, so let us see now. If we'll see the gene, or we see the DNA. We see the DNA. This is in the nucleus. Right now we are looking at the nucleus. So let me say this one is the DNA for the insulin. Okay, this one is the DNA. Okay, or the genomic sequence actually. This is the genomic sequence. Okay, the gene. If I say this is the 5' end and this is the 3' end. This part is an intron and this part is also an intron. So this is intron 1 and this is intron 2. We know that introns do not code for anything. These have just regulatory functions. We have 3 exons. Exon 1, exon 2, exon 3. Now exons do code for something. Exon 1 and exon 2 together, they code for untranslated part of mRNA. They code for the untranslated part of mRNA. The exon 2 alone code for the signal peptide and the B chain. The signal peptide and the B chain. Together exon 2 and exon 3 code for C peptide. Exon 3 alone code for the A chain and again the untranslated part of mRNA. Okay, so we have this 5' end is also, this is the cap site and this is the poly A site. Okay. Now we should remember this, that exon 1 is going to code for untranslated portion of mRNA, exon 2 is going to code for signal peptide and B chain, exon 2 and 3 together is going to code for the C peptide, and exon 3 alone will code for the A chain as well as the untranslated portion of mRNA. Before going on to the cycle, how it is going to form, going beyond the DNA into the RNA formation, let me tell you a few things. That insulin is synthesized as a part of a large pro-hormone. It is not directly synthesized as an insulin. There is a large pro hormone out of which insulin is synthesized. So actually it is, what we are going to synthesize is a large pro hormone. Okay, the gene for insulin, the gene for insulin hormone is located at, this is located at the short arm, this is located at the short arm of chromosome 11, short arm of the chromosome 11. Okay, so in the so this is the gene located. Okay, so I told you that insulin is formed as a pro insulin. Okay, then there is a 23 amino acid signal peptide. Once it enters into the endoplasmic reticulum, 23 amino acid signal peptide is removed and it converts into a proinsulin. Okay, this I told you exon 2 codes for the signal peptide. This proinsulin, once it enters the endoplasmic reticulum, 23 amino acid signal peptide is removed and the remainder of the molecule is folded. Remainder of the molecule is folded and disulfide bridges are formed. Okay, and this is known as proinsulin. So what is the difference between proinsulin and the proinsulin is 23 amino acid signal peptide is removed and it is folded and disulfide bridges are formed. It is folded and disulfide bridges are formed. Okay, the peptide segment here, if we see the peptide segment here, it consists of A chain and a B chain, and these A and B chain are connected together by a C peptide. Okay, C is the connecting peptide. So the A and B chain are connected together by a C peptide. Okay, C peptide will facilitate, this facilitates the folding, and once it is secreted, it is detached. The C peptide gets detached before it is secreted. The insulin is secreted, the C peptide gets detached. Okay, so I hope you people have understood this thing, that the insulin is synthesized as a proinsulin. Okay, and once it enters the endoplasmic reticulum, 23 amino acid signal peptide is removed and the remainder of the molecule is folded and disulfide bridges are formed, and this is known as a proinsulin. Okay, the proinsulin, the peptide has two chains, A and B chains. The A and B chains are linked together via the C peptide. So C peptide is the one which is facilitating the folding. Okay, and when the insulin secreted, before being secreted, the C peptide gets separated out, it gets detached. So if we see the beta cell, what it is secreting both the insulin as well as the C peptide. Actually, in a normal beta cell, it secretes 90 to 97% are what it is insulin. Okay, and equivalent amount of C peptide is also synthesized. Obviously, we know that there is a A chain, B chain. So one insulin molecule is equivalent to one C peptide. Okay. So how much ever insulin will be secreted, the similar amount of C peptide will be also be secreted. Okay. So this was the basic. Now we'll come back to the DNA where we were. We have talked about that there were 2 introns and 3 exon sequences. Okay, and this is a 1500 base pair. Okay. Now we go to the RNA form. It will form an RNA. Okay. This is around some 550 nucleotide, and this is in the cytoplasm. Now it has entered the cytoplasm. Now it will go into the endoplasmic reticulum, and we have heard that in the endoplasmic reticulum it is going to get folded, and it is going to form a proinsulin. We all know that signal peptide would have removed by then, so there would be a folded insulin. Okay, so this folded insulin will have disulfide bridges. Okay, so it will have disulfide bridges. This one is the A chain, this one is the B chain, and this is the C peptide. And we know that when it is going to enter the secretory granule, secretory granule, after going through the Golgi apparatus, packaging and all, it is going to enter the secretory granule, and the secretory granule, we have heard that then it will cause exocytosis in the secretion of the insulin. Herein, the C peptide is going to get separated. The C peptide is going to get separated, and there will be just an A chain and a B chain, and they will be connected via disulfide bridges. Okay, so we'll just have a A chain, we have a B chain, and this C peptide will be separated. And then we know that if this secretory granules membrane will fuse with the membrane of the cell, these two will fuse, and then the insulin is going to be exocytosed outside. Okay, so this is the synthesis of the insulin.